Hello students, welcome back in biology class. As you are knowing, the chapter heredity is going on and one introductory video I shared with you. Later on in this chapter, before that, the date is 9/10/2020. Subject is biology, class 10th, and the chapter is heredity. And in heredity, today we will study about the rules for the inheritance of traits. Rules for the inheritance of traits. Dear students, in human beings, the basic rule of inheritance is related with the fact that both the parents, mother as well as father, contribute practically equal amounts of genetic material to the child, and each child have traits of each child is under the influence of the DNA of mother as well as father. It means each trait in the child will have the two versions. One it received from its father, and second version it received from the mother. Okay. Gregor John Mendel was a scientist. Gregor John Mendel. Lifespan is 1822 to 1884. He worked about more than a century ago on this basic rule of inheritance, and he focused on contrasting characters. That means the characters which are visible and easily identifiable, like if we talk about the height character, and its two versions a person can be tall or can be dwarf, or a plant can be tall or can be dwarf. So this is the character. And if we make a parameter, a line of any parameter, from suppose I am taking 10 centimeter, so the person or the plant that has the height less than 10 centimeter will call dwarf, and the plant or animal or the person having the height more than 10 centimeter will call the tall. But in whole we are talking about the character, that is the height. So this I am saying that a child receive two versions of each character, and one version it received from its father, and second it received from the mother. Okay. Mendel worked on this basic rule. Gregor John Mendel worked on this basic rule of inheritance, and he worked during the year of 1856 to 1865. During this period, he conducted the experiment on pea plant and recorded some findings, and he published his findings in 1866. In the proceedings proceedings of natural history of society. Brunn is the place. Okay? In 1866. But unfortunately, his work remained unrecognized, unknown for a longer period of time. Okay? Longer period of time. Later on, three scientists are like Hugo de Vries, from Netherlands, Karl Correns from Germany, and Erich von Schemark from Austria. These scientists rediscovered the Mendel's finding in 1900 and republished in 1901, in the year of 1901, and then the Mendel works come in light, and the findings of Mendel form the basic of the science genetics. Genetics basics comes from the findings of Mendel, and because of this contribution, Gregor John Mendel considered as the father of genetics. Father of genetics. Okay. So this you have to know the basics and the work of Mendel. Later on, how Mendel conducted the experiment. Do you know on which plant Mendel conducted the experiment? Perhaps you will aware, some students are studying ahead in the chapter. So experimental plant of Mendel. Plant of Mendel, and it was pea plant, that is called the Pisum sativa. Pisum sativa. Do you know why Mendel chosen this plant? Mendel chosen this plant because this plant is easy to grow. Easy to grow. Second, having shorter life cycle. Shorter life cycle. Easy to grow means anyone can grow, anywhere it can be grown. Okay, and no special agricultural practices are required to grow the pea plant. Shorter life cycle enables the plants to grow more number of generations in given period of time. Okay, if shorter life cycle. Third, the bisexual flowers. Plant is bisexual. Four, the contrasting characters that can identify easily. Characters. Okay. So these are some reasons why Mendel. There are many other reasons also. why Mendel chosen the pea plant as the for his experiment. Okay. Now the next in the chapter is methods. What are the methods by which Mendel conducted the experiments? Methods, yeah, steps of Mendel's experiment. Okay. The first steps, there are, as you are knowing, the experiment has many steps. So Mendel's experiment has also some steps like pure line selection. Pure line selection. The first step. Pure line means the selection of pure line pea plants. The first process, first step. Pure line plants are what? The plants that shows, means the characters that appear shows same character in many generations. Many consecutive generations. Okay. That means any character, like if the plant is tall, then in further generations, for many coming generation, it is tall. Means the plant has observed for many generations to choose the pure plant. Okay. This is the first step. Then the second step was the cross breeding. This is also called hybridization. Cross breeding, hybridization. It means, suppose we have a character. It is pure tall, and this is the same character, that is pure dwarf. Pure dwarf. So these are the pure line plants. This first system. Then they are allowed to cross. Allowed to cross breeding, that is, it will produce, this is the hybrid, and this process is known as hybridization, and this hybrid is the result of F1 generation. Now let's understand the things. Crossbreeding means, that means the sexual reproduction, and the parents are different. Okay, so see, the parent is tall and the parent is small. It may be the female, it may be the male, or it may be female, it may be the male. Any? Okay. Now when the crossbreed, Mendel crossbred from tall pure line plants, the coming generation is having the characters of both the TT. See here, this tallness is the phenotype that is visible. Tallness is phenotype because we can observe the tallness, whether the person is tall or of short height, we can observe it. And this T, that is the code that is present on the gene, is the genotype. Means combination because of which this character appear. This is genotype. Okay, and the phenotype will be tall here also. This plant is first generation plant is also tall. All plants will be tall. Okay, and this phenotype is tall, this phenotype is tall, but the genotype of this tall is two different types, and the phenotype of this is the similar, that is the T in this. Okay, because a pair of characters are there. Now see here, this type of means the chromosome that has the same copy of character is called homozygous. This is called homozygous. Okay, and the chromosome that has the different copy of the character is called heterozygous. What it will called? Heterozygous. Okay, this is, I am writing here again. The first is the pure line selection. Okay, see here, capital T, capital T. So this phenotype of this is tall. Tall is what? Phenotype. This character can be observed. And genotype is this. And this is what? Itself is the genotype. Means combination characters present on the gene. Similarly, T small, that is the phenotype is what? Dwarf, and the genotype is TT. They are allowed to crossbreed and they produce in this way TT. So this is the F1 generation, and the characters in F1 generation, the phenotype is tall and genotype is heterozygous. It is what type? Heterozygous. Heterozygous. And these are homozygous. Means they will have the same copy of characters. So this is, so in F1 generation that produced by the Mendel is by crossing the pure line plants, and Mendel focusing on the character to choose the pure line plants. Now bisexual flower is how able to crossbreed, because self-pollination is the property mostly of bisexual flower. And how it will be cross-pollinated? So emasculation is the process. What in emasculation? Removal of gynoecium. You studied the gynoecium is the male part. Gynoecium from immature flower. The flower that is not mature. Okay, from that flower the gynoecium has been removed. This process is called emasculation. In absence of, and after removal of the gynoecium, the flower bagged, means covered, to protect the pollens of other plants fall on the stigma. Plant covered after removal of gynoecium. And afterward, when the flower become mature, the female part become mature, the pollen from chosen plant, the plant that is chosen as pure line, has been brought and dusted over the stamen of the plant, and in this way the crossbreed will happen to produce the heterozygous hybrid F1 generation. Now the third step, this is the second step. The third step in the experiment of Mendel is, third step is the self-breeding. Self-breeding. Self-breeding means asexual reproduction. No, it is sexual, but the pollens are not from the different types of flowers. Means we just produce the T type of in F1 generation. This is the product of F1 generation. They allow to self-breed between them. Okay. So then what the result will be? Pure line 1, then 2 hybrid and 1 pure recessive. Look here, 1 and this is 2 and this is 1. Means what is this? Genotype. Okay, and phenotype, these 3 are tall and 1 is dwarf. Okay, in this way. So this is the self-breeding to produce the F2 generation. This will be the F2 generation, second filial generation. Okay. So these are the steps in the Mendel's experiment. Mendel focused on the contrasting characters and the cross that done by the Mendel's cross based on one contrasting character was called monohybrid cross by Mendel. Monohybrid cross. And based on experiment, means cross based on more than one contrasting characters, if it is based on 2 characters, then dihybrid cross. If based on 3 characters, trihybrid cross. In this way, he conducted the experiment. Okay. Test cross is what? Test cross in which a plant of unknown genotype, is crossed with a plant of known genotype of recessive characters. Okay, so that is the test cross. Reciprocal cross, in which the parent's sex changes, then the original cross. Means in one class, suppose in original cross, if A is chosen as male and B has chosen as female, then it is the original cross, and B is chosen as female and A is chosen as male, then this will be the reciprocal cross, ulta. Okay, so this is all about today. Watch this video again and again, and soon we are coming in direct contact with you. Okay, till then revise the chapter and watch the video carefully. Thank you for watching. Have a nice day.